Okay, thank you very much, Sachin, for that introduction to Sati. So as Sachin mentioned, the focus today is on the cryo-electron microscopy part of Sati. Um, the NCR region has a very strong tradition in structural biology, primarily crystallography and NMR, but in the last few years, people have been moving more towards cryo-electron microscopy. And right now, we have a significant number of laboratories in the NCR region where the PIs have been trained. They have come as cryo-electron microscopists. And then we have colleagues who were crystallographers and NMR biologists now. To some extent, they're moving to cryo-EM. So today, we have a mixture of scientists who are interested in cryo-electron microscopy and the students and postdocs who are very happy to welcome. I hope you will benefit from these talks that we'll have 5 talks from now on. So the first speaker for us today is Dr. Prem Kaushal, who is faculty at the Regional Center for Biotechnology, Faridabad. Prem did his PhD from IISc Bangalore, then his postdoc at Case Western Reserve University and Wadsworth Center, which again has a very strong tradition for cryo-electron microscopy. And Prem's laboratory works on ribosomes. So he's going to talk about their latest research in this area.
Okay, good morning. I would like to thank Sati Foundation, especially Dr. Manideepa, for giving me this opportunity to present our latest work that we are doing in our laboratory. And today I will be talking about a story that is unique mode of mycobacterial ribosome hibernation that was revealed solely by doing cryo-electron microscopy. So ribosome, as all of us know, that it is a protein synthesis machinery. It has 2 subunits. The small subunit, which is the decoding center, where the A, P, and E site tRNA binds, codon anti-codon interaction takes place here, and the large subunit that harbors the peptidyl transferase center, and where the actual peptide bond formation takes place, right? So to function, to do the protein synthesis, the 2 subunit has to join together and form a functional ribosome that is 70S in prokaryote and 80S in eukaryote. But this protein synthesis machinery, it's a very fundamental process, and ribosome does in all cell, and it in nearly the 40% of non-antibiotic targets the different steps of protein synthesis, and it consumes nearly half of the cell's energy. It's energy intensive also. Now, problem, this is, okay, this is when there is a normal growth, everything goes okay, all is well here. But problem is when there is a stress, right? The bacteria has this lifestyle of feast or famine, right? Sometimes they have enough food, sometimes they are in stress. So they have evolved different way of adapting to this thing. First thing is they slow down the overall cellular machinery, including the protein synthesis. How they deal with this protein reduction in the rate of protein synthesis is different way. One is they have the alternative set of ribosomes. So they use different set of ribosomes. Second is some of the ribosomes they inactivate lock, and some of cases they switch to from leadered, where the mRNA has 5' UTR, to leaderless translation. So our lab's focus is on this. This process is called ribosome hibernation, where the bacteria temporarily inactivates the ribosome by reversible binding of hibernation promotion factor. Okay. So if we look into the literature, that this process in bacteria, including E. coli, is done by the ribosome modulation factor comes, joins 2 ribosome subunit, and hibernation promotion factor short comes, form a 100S. and the single RAI protein also inactivates that in 70s. And there is another form, that is the HPF long, where the two HPF, hibernation promotion factor, they will bind and the C terminal will form a dimer and form a 100S. If we look into the domain architecture, it is like this. RAF, rhizome modulation factor is slightly different, but it has an N-terminus domain. When it has it's kind of a dual domain, it forms 100S. If it is a single domain, it forms a 70S. What in Mycobacteria that causes tuberculosis has a different setup. That has a two hibernation promotion factor. Looks like this is longer form. Ideally, it should form a dimer, but it has a longer form that I will call it hibernation factor long in my subsequent slide. But it forms 70S. It doesn't form 100S. That was the question we want to address. What is the problem? Why it cannot form 100S? What is the advantage, disadvantage, etc.? Why Mycobacteria have evolved with this kind of mechanism? Okay. So we started with the RAF, and it's some 30 kilodalton protein, and it's regulated by DOS R regulon. So when there is a hypoxia, this DOS R regulon overexpresses RAFH, including some other 48 genes. And in the hypoxia, it is shown that RAFH is the sole responsible for the survival in hypoxia. Okay. So then obviously we started the cryo-EM. For that we need a ribosome. My student Neeraj and Shivani, they purified the ribosome. And this is the 70S. This is a sucrose gradient ultracentrifugation after fractionating. You see this peak, and you can see the both ribosomal subunit are there, ribosomal rRNA. This is 50S, 30S. Problem was we tried, but we failed. If we use this 70S as such, because there are factors that gets co-purified, and your factor, whatever you want to make a complex, it didn't work. So we use the second strategy. We reduced the magnesium concentration and dissociated 70S into its subunit 30S, 50S, and then use these 50S for our study. This is the assembled one. Okay. Now, then RAFH was purified by affinity and gel filtration chromatography. Then we use sucrose pelleting assay. That is actually you make a complex, put on a 50% sucrose gradient. If it is bound to ribosome, it will settle down. You will find your RAFH protein with the ribosome. If it doesn't, like here in control, you don't see the intensity of the band. So we were sure that this complex is there, and we did some inhibition, and we found that it is inhibiting the protein synthesis. It means the protein is active and good to go for EM. Then we did a screening that we do at our RCB ATPC facility, and confirmed that these ribosomes are uniformly distributed, RAF is there, everything is almost certain, and sent data sampled for the National Cryo facility at NCVS Bangalore, and we got okay. Now here I want to go back for the students' sake, give the little bit idea about how this EM has evolved and what is the principle behind this. This is important to know, especially for the student. Okay, so the first cryo-EM reconstruction was done by David DeRosier and Arun Klug in MRC laboratory in 1960. What they did from 2D of this bacteriophage T4 tail. For this, they took one picture because it has a 32-fold symmetry, and you could use this and generated the first 3D from 2D projection. So this ideally is the first EM reconstruction. Okay. Now how they come up, it was very based upon the very simple thing. If you see the EM images, the 2D projection we get, the EM images are the 2D projection of 3D object. This is 3D object, our EM images is the 2D projections, right? And they come up with this theorem. This is the all one should know in cryo-EM how the from 2D to 3D it comes. It's a very simple technique. Basically, it says this is called projection theorem. You suppose you have a 3D duck, right? You put that duck in a microscope, but don't put a duck in microscope. Okay, and what you will get? You get a 2D projection of that duck, means 2D projection in different orientation, and you do a Fourier transformation. So in Fourier transformation, it will be a slice. Here it is a projection, it's a slice. And you have a 3D duck, that is your model, initial model, and do a computationally make its 3D Fourier transformation. And these 2D slice will be always a central section. These 2D Fourier transform, these slices will always be a central section in your 3D Fourier transformation. Okay, this is the concept behind the EM image. That whatever we get 2D images, then we do the 3D only. Fourier, this projection theorem is involved, nothing much. So okay, now we got this data, and it's a quite huge data, 12 lakh particles from 12,000, nearly 12,000 micrographs, 2D projection, 2D classification, and then 3D. We selected from 2D some 7 lakh particle, and we got tRNA, RAF, MT, RAF and tRNA. But problem is ribosome, it's a giant, and good thing is it's easy for the alignment, but bad thing it comes with a lot of heterogeneity, that is the conformation and composition heterogeneity. Conformation means one ribosome is different from another, right? Another is composition, because we are looking for RAF, and its occupancy may not be 100%. I will come to know, I will tell you how we sorted this thing. So for that you have to use different strategy. That the strategy that worked for us was we first did the 3D classification and selected which particle as a RAF and RAF-E. And then we focus classification only on the small subunit, because small subunit is the RAF binding site, okay? And further sorted and reached to 1,53,000 particle. There 36 was absent and was empty. Here it has a particles, okay, a RAF. And to do further, to improve our data, we did advanced refinement, that is CTF refinement and particle polishing. Basically, initially when you get a movie stack, you align this stack based upon a small segment, small pieces of that segment. But at this stage, you are picking each particle and looking at how it has drifted throughout its movie. Suppose you got a 25 movie frame. You are taking one particle and figuring out how it has drifted and applying correction back for the each and individual particle. That has helped us to improve resolution, not much, but the quality of data was improved very much. That's important, right? Now another problem was that because RAF NTD was fine, CTD has a problem. So we used, this is called signal subtraction, and you just focus on this region where your particle is, and subtract this as a signal and consider this everything outside as a noise. So you will have only data for this. Is a computational you can do, and it is available in Rely on 3, and then we classified different things. What we interestingly found is in one class, this is RAF-CTD, it comes with the S1 protein. This is only RAF-CTD, this is RAF-CTD and tRNA. So we further classified, okay. Now then obviously our focus is CTD, we want to add more particle. So one thing we merged all things, so that gives a CTD better resolution. And in one class only the in one class only the S1 and another class only the tRNA. Okay, this way we sorted this thing. To understand more about this, this is called multibody refinement, where you treat each subunit of ribosome as a different body and let them freely align and move. Okay. Now to understand more about, I recommend for the, especially for student, you read this. This is Shore's paper that explains how this signal subtraction actually works. Basically, suppose you have a volume and you want to focus only on this region. What you can do, you can theoretically apply a mask and divide into two. This will be the surrounding region, this is your the region you want to focus. And suppose this is an experimental image. Now you can subtract this noise, or you can say that this is the region outside your signal, like this. And if you subtract the outer region, it will become like this. So the region what you are focusing outside is almost close to the blend. That helps in better alignment. Obviously, this image can align better to this image compared to this image. This is how the signal subtraction works, and it successfully was done by Shore's group in 2015. Subsequent, a lot of people have used. Similarly, for this multi-body refinement, originally it was done for spliceosome, because spliceosome is notoriously heterogeneous. To sort all these populations, they systematically did multi-body of head, body, foot, and came up very high resolution of each. So these are the two paper I recommend for, especially for the student. Then once we got this is the way we estimate the resolution. It is a Fourier shell correlation at 0.453. That is your resolution cutoff, and one should definitely look how the local resolution looks. What you are reporting in this, it is a overall. That may not give you the right feeling. So this local resolution here, it says the starting range is 2.5, and we could see most of our resolution data is 2.5 to 3.5 angstrom resolution. Okay, so we are certain that everything is okay. This is what the structure looks like. Small subunit that I said before, a large subunit, and this is NTD here. And if I rotate this molecule roughly by 180 and little tilt it, it looks like this. This is called the exit site, where this is E site tRNA, and our CTD is bound here. It has S1 protein and the S2 protein that is actually sandwiching this thing. Okay. Now, in this, it's 1,53,000 finally. We started with 12 lakh and ended up with 1,53,000. Yes, not weeks, it's a 3 session, 3 into 2 days, 6 days in NCVS National Cryo-EM facility, yes. Yeah, our facility is for screening, we do only screening, yeah. Okay. So this is what NTD, if we take this, you could clearly see the secondary structure feature. See, resolution reporting one thing, if you say 3 angstrom, if you don't see side chain, it means that's not true, it is something overfitting. You could clearly see secondary structure features, alpha helix, beta sheet, separated beta sheet. Okay, this gives the confidence that what you are reporting is right. And this is further, if I take this piece here, this is linker and anti-shell Dargano sequence, and you could see clearly resolved side chains, and there is no ambiguity. It's kind of a crystal clear, right? And yep, further, yeah, this is a movie. I hope it will work. Yeah, it's not only 2-dimension, but you can see in all dimension, every side chain is very much clear. Then if we dissect this NTD and its interaction, how it is interacting here, 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 each and every side chain amino acid very much clear, no ambiguity. So this is the power and the beauty of the cryo-EM. I think this is the right time, golden opportunity. Students here, they should grab this opportunity. That's what I can say. Okay, here also what I did, I put, there is a movie for everything here, but when I was doing checking, it didn't work because it's, let me try if it works. Okay, just I want to convey that it's like crystallography. Eh? Oh, thank you, thank you. Okay, I think it's enough. I think I conveyed the message, right? That's more important. Yeah, now this was about the NTD. Now coming to the CTD, CTD is here, sandwiched between the S1 and S11 protein. Because CTD is still dynamic, we couldn't get as good resolution as to the NTD. So but we can clearly see the features. And if you look, this is something that first time we saw that we figured out, we showed that S1 is involved in the hibernation. And clearly you can see it looks S1. This is S2. This is this region fitted. This region fitted, and this alpha fold gave us this structure, and 2 domain they are nicely docking. Okay, now so this is the thing. If we see that CTD, it is little longer in our structure in RAF, and the other reported structure where they reported at hibernation factor long, where I said the 2 CTD forms a dimer. That structure looks like this, but in RAF it is already dimer. So there is no scope it can further dimerizes. So that was the reason it cannot further dimerizes, so it cannot hold to 70s, so it cannot form the 100s, right? So that is what structure has revealed, that this is the basis of not forming the dimer. Okay, this is how it looks overall. And interestingly, when we docked our structure on a initiation factor, means where the ribosome initiation is just about to start, and what we found that this is NTD, linker, and CTD. NTD bind to the decoding center. Obviously, it will block the translation because the P site tRNA will not bind. Linker bind to the anti-shell Dalgarno sequences. So it means that is blocked. So there will not be the shell Dalgarno, anti-shell Dalgarno sequence interaction. mRNA will not be recruited. But there is a weak anti-shell weak shell Dalgarno sequence present on the mRNA. It use this S1 protein that I showed, and for the recruitment of mRNA, that is blocked by CTD. Right? In bacteria, 25% of mRNA are leaderless. So their translation starts directly from P site. That is also blocked. So this RAF-H has a strategy to block all kind of translation initiation. Okay. And these are the interacting, where it interact on, this is a 2D projection diagram of 16S small subunit 16S RNA. And it interact to anti-shell Dalgarno, this region, And anti-shelled organo sequence or 3' end of 16S is prone for the RNA degradation. So it is protecting that also. So it is one way it is protecting from degradation, another way it is stopping, not allowing to initiate the translation. So that is the perfect way of preserving ribosome, right? A 16S RNA, it comes in the closed form. That's exactly when what it adopts when initiation starts, when initiation factor binds. When there is not mRNA is not bound, it is open form. Okay. No, we compared the already structure because we don't have the initiation complex structure that is already reported. So we use that structure. Okay. So basically, if we look, this is the RAFH from MTV, and it is the CTD is longer. So that domain is kind of duplicated. That's why. But in other long form of RAFH, this CTD is shorter because it has only one CTD, but it RAF has a repeated CTD. That's the why it is already in a dimer form. Okay. I think I have only few slides now. Now it comes to how to understand the why it cannot form a dimer. It is obviously okay. It is not forming. It's already dimer, but why it cannot form a 70S, a 100S? Simple thing is this is the situation in our case. NTD, CTD, helix 54, S1, S2, and if we look a dimer interface or 100S interface of other bacteria, this is a CTD, NTD and CT position is different, right? And when we docked our structure into the 100S structure, this is a simple exercise, right? And what we found is it's making huge steric clash. Helix 54 is clashing here, CTD is clashing with helix, another helix here, like that. So it is not possible. But this arrangement in other bacteria of forming 100S is these two CTDs coming and dimerizing and holding together, right? That gives a structural basis of its interpretation. So we basically showed one more mode of ribosome hibernation, that is, even though if you have a two domain hibernation promotion factor, because its CTD is already dimerized, it cannot further dimerize, it hibernates in 70S form, but here the longer form, they forms a dimer, so it forms a 100S. Another mode of this is in literature, this is also in literature, and this is also in literature. This we had added up new information kind of, okay. Yep, and it not only blocks the initiation or RNA degradation, because lot of antibiotic binding site overlaps with the RAFH NTD binding site. It's a very systematic strategy bacteria has developed, that these antibiotics will also become inactive. Okay. With this, just I put one slide here. We also have a 200 kVA electron microscopes facility. It is basically used for the screening because it's a 200 side entry mounted holder. Yep, you may think of using this also. It is in ATPC, and you can write it service at the rate of ATPC RCB.in. Okay. And with this, I would like to acknowledge my student. I have a small group, Neeraj, Shivani, Ankita, Somen. All are really hardworking, very brilliant students. I really thank them. And I thank Dr. Nair for all advice or all guidance. Todd Gray for giving the clone. ITPC facility, Reena and Madhav for helping us in EM. Especially thank to the Binod and Sucharita from NCVS. They collected very good quality data. It's a quality data gives you a quality structure, right? Okay. Thank you very much. Would be happy to take questions.
Are there any questions?
Prem, it was indeed a very nice talk.
Thank you.
So my question is so far a lot of crystallography and cryo-EM structures analysis of ribosome have been elucidated.
Yeah.
So briefly, what is the new information that you have, which in your view could potentially add to understanding the biology of ribosome better?
Okay. See, ribosome, if you see the structure, and the Nobel Prize went for the crystallographer, right? Benke, Yadayonath, and Tom Stites. But the number of structure from X-ray was limited, because it's only that one could crystallize, you will get the structure. And in EM, it's easy that you make different complexes and you get structure. Not only that, within one data set, people have reported even 20 substructures at different stages of its activation. So this mechanism, see, as a structure it was there, everything was there, but the mechanism of understanding at different snapshot, that has helped cryo-EM. Cryo-EM has helped that. Yeah, yeah, student, yeah, definitely.
Thank you, sir, for the great talk.
Yeah,
I have a few questions regarding sample preparation.
Sure.
So you said that you used sucrose pelleting. So I wanted to know, did you use a ultracentrifuge for that?
Sucrose pelleting is for doing a assay, because we are forming our complex with ribosome. If it is bound with ribosome, then ribosome pellet down in the high speed centrifugation, the factor will also come. If it is not bound, factor will not get settled. So that is for the assay. Sample preparation has nothing to do with sucrose gradient.
So, sir, how did you initially isolate the ribosome? Did you use any chromatography techniques?
No, for ribosome isolation, the protocol are almost standardized. You do a sucrose gradient. There is a pelleting is different. Gradient is you use 10 to 50% gradient. Based upon the Swedberg constant, the different subunit will settle in that gradient in ultracentrifugation. Then you fractionate it and absorb the peak and collect that peak and do the thing.
So, sir, is there any kind of contamination from membranous proteins or vesicles observed in case of sucrose gradient?
Membranous, see, in ribosome, they are also associated to ER. So we also get endoplasmic reticulum contamination. What we do first 2 rounds of purification. One is crude, then we do homogenization, extensive homogenization and centrifugation. That has helped us to get rid of membrane, and sometimes detergent also helps.
Okay, sir. Thank you so much, sir.
Yeah.
The one which is not seen in the other ribosome structures, one structure where you use the alpha fold model.
Okay, alpha fold we used for to find out the structure, because we don't because
so this this protein was not there in other ribosomes.
Yes, this is the first time we showed that S1. We clearly showed. Before there was a lot of controversy. Some were believing that this is S1, some were not, because their resolution was poor. This is the first time we showed that S1 is involved in stabilizing the hibernation promotion factor, because here the resolution is somewhere 5 angstrom. You cannot build ab initio, right? So otherwise you have to do secondary structure build, then loop build. Better is we have alpha fold and take it and dock it.
So you only the alpha fold gave only the domain, or even the loop and other structures?
No, alpha fold gives you everything. That's the problem.
Does it match with everything?
That's the problem. So essentially, see, this is what we got from alpha fold, like this. And obviously these 2 domains were matching, and we docked them in this here. Other, it means they are disordered. So it is just for the docking alpha fold. Yeah.
And you showed that a lot of local regions have higher resolutions.
Yes.
So did you achieve higher resolution in some local structures to try and see these modifications on whether they are changing the local structure of the RNA
See,
or the ribosome?
our is from 2.5 best to 3.5, but actually if you want to see modification that people have shown, you need like a 2 angstrom and some 2.1 angstrom. So we didn't attempt to find out. But with cryo-EM 2 angstrom ribosome structure, people are showing lot of post-translation modification. It's very much evident, means you can clearly see.
So any thoughts on how to increase the resolution in these local substructure regions?
No, see,
Or just you have to
we
collect more
can,
images?
I mean, the strategy we use for RAF, we can use for that particular region. But we knew that we are going to have the best 2.5 angstrom resolution because our pixel size is 1.07 angstrom, and best theoretical resolution we can achieve is 2.12, sum of 2 pixel. So 2.12 angstrom is there, we will never, and there will be some, nothing works perfect, right? So if we want to achieve high resolution, we have to go to higher pixel, higher magnification, like 0.83 magnification. That we have not thought because
Okay.
maybe in future, yeah.
Thank you.
Good question. Nice question. Okay, there is a question. Thank you.
You have refolded the alpha fold structure into your model. So about the sample preparation, if like we are working in cytoskeleton proteins, especially the myosin, so while the purification from any source organism, most of the time like the heterogeneity of myosin isoforms comes.
Yes.
So do you have any suggestions how we can overcome that?
One way is you collect lots of data, and then I know that in India it's not that easy to get a lot of data. And then keep doing classification. That is the one way. Another way is find out that there are some buffer or some component that stabilizes, right? If in literature, if in myosin, other people how they have done.
I have referred to the literature, sir, but the problem is most of the two motor domains of the myosin protein. So while the particle analysis, it will be really difficult to separate that which isoform of this myosin is. So like during the density gradient centrifugation, most of the myosin isoforms comes along with the actin. They are already bound, and the structure which has published through cryo-EM, they are bound with the actin. But I wanted to specifically know, like, is there any method to separate out the between those isoforms during the purification or not?
Purification, you are using sucrose gradient, right?
Yes.
Varying the sucrose gradient length, that can help.
Okay. And
second was?
Second was about when we have modeled our myosin structure in the alpha fold. So the motor domain structure was quite formed well, but rest of the other domains in the tail region were not, they were unstructured while during the production. So how we can use that alpha fold structure for superimposition and look at the electron density if we can get it from the cryo-EM images?
No, see, alpha fold is just to dock in your EM density.
Okay.
Alpha fold is not going to help you to improve the EM density.
Okay.
Right? To improve the EM density, you can use signal subtraction, multi-body refinement. That helps.
Okay. Thank you so much.
So you talked about signal subtraction. So what was the particular advantage in terms of resolution or like computational speed?
You are making equivalent to noise.
Yes.
So you are dampening the background. That is not actually background, but signal subtraction is a background. So you have a better alignment.
Okay.
Computationally, it is not expensive. It's very not expensive, I will say. It works well. Yes, but you have to make different attempts. It may not work one shot that you subtracted and it worked. So that is there. Yeah.
Okay, thank you.
Okay, so I think in the interest of time, we'll move on to the next speaker. Thank you very much to Dr. Kaushal for an amazing talk.